Um, okay, welcome everyone to Infex Therapeutics webinar on their lead phase 2 respiratory drug candidate RESP-X. My name is Lewis Jepson. I'm a senior associate at Walbrook PR. We are media advisors to Infex Therapeutics, and we're hosting today's webinar on their behalf. So today they're going to run through a quick presentation on the candidate RESP-X, and there'll be a short period after the presentation for a bit of Q&A. I would ask you can submit your questions during the presentation. Please use the Q&A function at the bottom, and the Infex team will get to those questions at the end. Any questions that they don't get to, we can potentially answer after the event. Without further ado, I'll hand over to Peter Jackson, CEO of Infex Therapeutics. Thank you, Pete.
Okay, thank you, Louis. If you could put the slide deck up for us now, and welcome everyone to this webinar on Infex's lead candidate in clinical trials, RESP-X. Can I introduce as well Vicky Savage? Vicky is our Chief Scientific Officer at Infex, and she'll be telling you about the disease and the mechanism of action of RESP-X, and also the excellent preclinical data that we've been able to generate. Then Vicky will hand over to Professor Colm Leonard, who's Infex's Chief Clinical Officer, and Colm will take us through a bit more about the clinical aspects of the disease, but also the excellent progress that Infex is now making with the RESP-X program, which is now in phase 2, so clinical stage asset. So next slide, please. So RESP-X is an innovative clinical stage antivirulence antibody, and we believe it has blockbuster potential in a specific disease area called non-cystic fibrosis bronchiectasis, NCFB. And these are chronically infected respiratory patients with a very poor prognosis. So we'll tell you all about that. And first of all, I'd like to hand over to Vicky, who will take over for the first part of the presentation. Thank you, Vicky.
Thanks, Pete. Hello, everyone. So if we could just skip to the next slide, please, and kick off. So yes, today I'll be telling you about our preclinical package of data for RESP-X. So RESP-X is a novel monoclonal antibody that specifically targets the bacterium Pseudomonas aeruginosa. And we're developing this antibody as a maintenance therapeutic for non-cystic fibrosis bronchiectasis, or NCFB for short. So this is a chronic lung condition that has well-defined diagnostic criteria. It is characterized by a chronic cough, the production of excess sputum in the lung, and patients often suffer from recurrent respiratory tract infections. Patients also have irreversible dilation of their airways, and the infections that they suffer are frequently caused by the bacterium Pseudomonas aeruginosa. And this organism serves to drive neutrophil activation, inflammation, and it causes progressive lung damage and exacerbations. Now, exacerbations are effectively flare-ups in the patient's symptoms. So for a period of time, they experience a sort of worsening of their symptoms. This could be shortness of breath or excessive coughing. And these exacerbations often require hospitalization and also antibiotic treatment. In severe cases for patients, they can experience multiple exacerbations a year, up to 12 in very severe cases. And the antibiotic therapy that they are given is often ineffective, as Pseudomonas is a highly resistant bacterium. So in getting that chemotherapy, it only really serves to worsen the antibiotic resistance that these patients suffer. So NCFB is often characterized as a sort of vicious vortex. So they have repeated bouts of infection and worsening lung damage, which only sort of exacerbates their predisposition to infection. So initially, the condition of the lung predisposes these patients to Pseudomonas colonization and infection. And once Pseudomonas is in the lung, it produces exotoxins, which serve to attack the immune system and really disable the infection clearance mechanisms present in the host. The exotoxins cause neutrophil degradation and inflammation and lead to progressive lung damage, which further predisposes the patients to repeated rounds of infection. So with RESP-X, we're really aiming to break this infection and lung deterioration cycle. And it's also worth saying that at this point that RESP-X is not an antibiotic, so it is not susceptible to antibiotic resistance mechanisms, nor does it drive the emergence of antibiotic resistance. And really through use of RESP-X in this patient population, we really hope to reduce the reliance on broad spectrum antimicrobial chemotherapy. Next slide, please. So a little on the mechanism of action. So as I said, this is a monoclonal antibody, and it targets a key virulence factor in Pseudomonas aeruginosa called the type 3 secretion system. So we've just got a cartoon here on the right-hand side showing this multi-protein complex. So it forms a needle-like structure on the surface of the cells. So RESP-X binds the end of the needle-like structure, a protein called PCRV. And in doing so, RESP-X actively blocks the extrusion of exotoxins by this pump into host cells. So RESP-X, in binding the system, reduces tissue damage mediated by exotoxins and also neutrophil degradation and inflammation. And collectively, RESP-X is able to restore the killing of Pseudomonas aeruginosa by the immune system neutrophils, and this better helps elimination of the organism from the site of infection. This mode of action has been confirmed by advanced techniques such as X-ray crystallography and immunohistopathology and advanced microscopy using ex vivo samples from lung models. We've also established some nice proof of concept efficacy data for RESP-X in preclinical models of Pseudomonas lung infection, which I'll come on to in just a moment. Next slide, please. A little more on the mode of action. So on the top left-hand side here, we can see an electron micrograph image, and the black dots in this image are RESP-X, and we can see the end of a Pseudomonas aeruginosa cell and the type 3 secretion system needle clump complex projecting out of the cell. So really we can see here nicely RESP-X binding the end of this system. And the mode of action is further summarized in the cartoon below. So the Pseudomonas cells are shown in orange, where they are primed ready to deliver exotoxins into the host cell. So the type 3 secretion system is shown in green, and we can see it making contact with the cell membrane of a human cell. So RESP-X binds the ends of the type 3 secretion structure and prevents the delivery of those exotoxins into the host cell, preventing host cell death. So there is 4 main exotoxins delivered by the type 3 secretion system: XO-S, T, U, and Y. XO-T and XO-Y are found in the majority of Pseudomonas aeruginosa strains. XO-S and XO-U, whilst not quite mutually exclusive, it does tend to be an either-or situation. So Pseudomonas will express either one or the other. And this is true in the table of strains that we've presented below here. So these are some of the bacteria that we hold at Infex, and we've characterized the exotoxins present in these organisms. So we've got organisms from lab origin that we use frequently, from cystic fibrosis patients, and also from non-cystic fibrosis patients. So we can see XO-T and XO-Y present in virtually all of the strains we've characterized, and the strains tend to produce either XO-U or XO-S. Next slide, please. So our partners on the RESP-X program, Shionogi, have done some really nice mode of action studies. So they recently presented a nice poster characterizing the mode of action in vivo in models of Pseudomonas lung infection. So I've just taken a snapshot image from this poster here. So effectively, what they've done is taken samples from the lung in a mouse model of Pseudomonas lung infection. And then using immunohistopathology, we have stained the Pseudomonas cells green, the exotoxin XO-U in blue, and also RESP-X is shown in red. And really what we can see as we go from no drug and then increase the dose of Resp-X from 0.015 mg per kg up to 1.5 mg per kg, we see a dose-responsive reduction in the amount of exotoxin delivered in the lung. Next slide, please. We've also taken a look at the diversity of the target protein, PCRV. So the pie chart on the right-hand side shows the sequence diversity in around 500 Pseudomonas aeruginosa samples, which have been isolated from patients. And the majority of these Pseudomonas bugs were type 0 or wild type in their PCRV sequence. And then a further 35 sequence types were found for this protein. But regardless of the sequence type, Resp-X was able to bind the target in greater than 99% of these organisms which have been characterized. We're also amassing a collection of Pseudomonas samples which have been collected specifically from NCFB patients. And this is in collaboration with the Liverpool School of Tropical Medicine and also Imperial College London. And encouragingly, all of the bacteria that we've screened so far have contained the PCRV target, and that the sequence of this target is consistent with the organisms which have been screened already in the pie chart on the right. Next slide, please. So on to some efficacy data. So we have quite a wealth of efficacy data for Resp-X. So perhaps the most relevant is our chronic infection model. So this is the image shown in the center of this slide. So effectively, what happens in this model is that Pseudomonas aeruginosa is allowed to colonize the lung of the mice for around a week. And then an exacerbation is mimicked by the use of cyclophosphamide. So this effectively depletes the immune system of the mice. And then the outcome is measured in terms of survival. So what we can see using Resp-X, we can see a nice dose-responsive efficacy with 100% survival of the mice being achieved at 1.5 mg per kilogram. We also have a variety of other efficacy data, particularly in acute models of Pseudomonas lung infection in mouse, and also a gut-derived sepsis model in mouse as well. Regarding safety and DMPK data for our preclinical package, we have pharmacokinetic information in 3 species for Resp-X, including mouse, rat, and monkey. And this data has demonstrated dose linearity in these species, and it's also enabled us to predict the human half-life of 20 to 40 days. And this will be ideal for infrequent dosing as a long-term chronic preventative therapy. The glycosylation profile for Resp-X is acceptable for therapeutic use, and preclinical toxicology studies have really demonstrated that we have a large safety window for therapeutic dosing in humans. Next slide, please. So just a brief summary for the preclinical package. So as I mentioned, Resp-X is a monoclonal antibody that targets a key virulence factor in this priority organism, Pseudomonas aeruginosa. The preclinical data we have suggests that it's extremely safe and well tolerated in preclinical studies. Using a chronic lung infection model to best represent NCFB, we've demonstrated protection in this system using Resp-X. And the PK and efficacy data we have in our preclinical package really support the infrequent dosing that will be required of a therapy to prevent exacerbations caused by Pseudomonas in this chronic lung disease, NCFB. And finally, as I mentioned previously, Resp-X is not an antibiotic, so it has the benefit of not being susceptible to antibiotic resistance mechanisms, and it does not drive the emergence of antibiotic resistance. And we are hopeful that use of Resp-X in this patient group will really reduce that reliance on broad-spectrum antimicrobial chemotherapy. So with that, I will hand over to our Chief Clinical Officer, Professor Colm Leonard, who will walk you through our clinical progress and clinical plans.
Right, thanks very much, Vicky. Moving on to a clinical focus, I thought it was helpful to start with a patient story. This is Scott Story from the British Lung Foundation, young man who'd been very active, and then age 40 started to develop symptoms of bronchiectasis, developing lung infections, ultimately becoming colonized with Pseudomonas and having very significant reductions in his lung capacity. And exercisability. And with these infections, they became increasingly more severe, requiring hospitalization for broad spectrum intravenous antibiotics. And the difficulty also being that it's not just about having antibiotics for Pseudomonas, but they tend to become less effective, these antibiotic courses. And again, these exacerbations due to Pseudomonas have massive impact on his work life, his home life, and friendships because of the time away from all of those areas, and a really very, very significant clinical impact. Next slide. So non-cystic fibrosis bronchiectasis has a well-defined severity scoring system that incorporates various factors, clinical parameters, radiological parameters, and also microbiological features. And this scoring system is called the Bronchiectasis Severity Index. And this has been very well validated across a European cohort of patients called EMBARK. And really the point of this slide is a couple of main things. Firstly, that this severity index is an excellent predictor of mortality and hospital admissions, and Pseudomonas colonization forms is a key contributor to scoring highly on that severity index. And those patients with severe disease who score highly, many of them with Pseudomonas, have up to a 30% mortality at 4 years, which is really quite dramatic, and up to 80% hospitalization rate, again, emphasizing the impact of Pseudomonas in this chronic lung condition. Next slide. This just talks about the colonization rates, again, across various parts of this European network, EMBARK. And while there's a range of colonization rates with Pseudomonas in bronchiectasis patients, overall, something around 25% of all sputum samples are positive for Pseudomonas. So a significant proportion of these patients are colonized with Pseudomonas. Next slide. So this is moving on to some US data, and really this emphasizes the large burden of non-cystic fibrosis bronchiectasis in the United States. And the graph on the top left-hand side shows prevalence and incidence, and then the graph on the lower left shows age-related prevalence and incidence. And you can see really on this left side of the graph that the disease disproportionately affects women, and also is more disproportionately affecting people as they get older. And the impact of these Pseudomonas infections in non-cystic fibrosis bronchiectasis patients has been well characterized, even in terms of costs in the US. And that's the graph on the right-hand side, that you can see that the incremental cost associated with somebody with this lung condition becoming positive for Pseudomonas is in excess of $31,000 per year for an individual patient. And you can then extrapolate from that to several billion potential market for RESP-X. Next slide. So this shows the exciting results from the phase 1 healthy control study that has now been completed here in the northwest of England. So we carried out this phase 1 study in 32 healthy controls, four different dose cohorts, and placebo patients in each of those cohorts. And what's fantastic is that the drug has a very clean safety profile, and moreover, not only were there no significant adverse effects at any of the 4 dose levels, but the pharmacokinetics really was as predicted from the preclinical data that Vicky presented earlier. And we showed in this completed study a half-life of over 30 days with dose linearity also. And this work was part supported by an ICON grant support. And the outcome of this phase 1 is that we now know that the dosing that would be needed for doses at 3-monthly intervals would be 10 milligrams per kilogram. And if we were going to dose at 2-monthly intervals, 6 milligrams per kilogram. Next slide, please. And now on the top left, so we're moving on to the clinical stage. So we move beyond the healthy volunteers. The top part of this panel on the left-hand side refers to the phase 2A, so the first study in actual non-cystic fibrosis bronchiectasis patients. So we're currently recruiting for this with something like 26 patients in screening for this part of the study. Were 2 cohorts of 6 that would receive 2 different dose levels of Respiratory and then undergo bronchoalveolar lavage to document both the primary endpoint of safety, but also to document that the drug gets into the lung and to look at the pharmacokinetics and make sure that the pharmacokinetics in patients is similar to the pharmacokinetics in the healthy controls. And obviously that phase 2A study that's recruiting currently will then inform the finalization of the plans for phase 2B, which we're well advanced already with those plans, with lots of support from key opinion leaders around the world. But that phase 2B in the first instance would be in 5 centers, main centers in the UK, where we would dose non-cystic fibrosis patients who are colonized with Pseudomonas with Respiratory at 3 monthly intervals for a total of 4 doses, with the primary outcome being looking for a reduction in the rate of exacerbations associated with Pseudomonas. So we have the drug substance available for this, and as I said, once we have the phase 2A data, we will do our final submission for approval to start the phase 2B study. Next slide. So really to summarize where we are in humans and in clinical stage, we know quite exciting that we have a very clean safety profile in human subjects with a long half-life from healthy controls. We're really excited to have started recruiting for that phase 2A study that will prove that the drug gets into patients, into the lungs, and also has a similar half-life, we think. The aims, of course, are of the phase 2A are that lung deposition and pharmacokinetic data and exploratory biomarkers to finalize the phase 2B plans. And as I said, we're very well advanced with those plans for phase 2B for that dosing over 12 months to show reduction in frequency exacerbations. And these phase 2B plans have been widely shared around the world, and we have strong interest from key opinion leaders, both in the US, Europe, UK and Ireland, and Japan for this work. And we also have strong patient support from the Bronchiectasis Advisory Group of the European Lung Foundation. So in summary, really, with the clinical phase of this project, we feel we have significant potential to address a really big unmet need in a really debilitating chronic lung disease. And so I'm going to hand you back now to Dr. Pete Jackson, the CEO, to talk you further on this topic. Thank you. Pete, you're muted.
All right, thanks. Thank you, Colin. Thank you, Vicky. Can I just remind everyone that if you've got questions for Vicky or Colin, please pop them in the Q&A that you'll find at the bottom, and then Carl from Infex will work through those and pass those over to us to answer at the end. So, having heard about the brilliant science and the technical side, clinical side, the great clinical progress that's been made, I'd just like to talk a bit more now about the commercial opportunity that we see in front of us with Respiratory in NCFB. We genuinely believe this is a multi-billion market, as Colin has outlined, with the number of patients and the cost to the health system of NCFB, and in particular Pseudomonas colonization in those patients. So summarised here is just the view of the number of NCFB patients that are currently characterised. So between the main European 5 countries and the US, there's getting on for a million patients, NCFB patients, and a further 5 million in the Asia Pacific. So potential global market in those major markets of getting on for 6 million patients. And as Colm outlined in his presentation, the incremental cost of Pseudomonas infection in those patients annually is over $30,000 from the US data, and 25 to 30% of NCFB patients are colonized by Pseudomonas, which is the target for the RESP-X preventative strategy. So overall, that represents certainly a $1 billion opportunity per annum for RESP-X commercial sales. Additionally, there's a widespread view that bronchiectasis is under-detected, and in particular, a recent study found that up to 9 million COPD patients in the US may also be suffering from bronchiectasis, and many of these with Pseudomonas colonizations. And similarly with asthma, over 600,000 potential asthma patients also may be underdiagnosed with bronchiectasis. So again, bringing it back to what Colm said, what we always have to remember is the patient impact. And with up to 30% 4-year mortality and the 8% hospitalization rates for patients, they desperately need a new drug to be able to reduce the impact on these patients and their loved ones. Next slide, please. So because of all of that, there is new and emerging pharmaceutical industry interest in NCFB therapeutics. And Infex's RESP-X program is actually leading the way in the antivirulence approach to breaking what Vicky described as this vicious vortex. So Insmed and Boehringer Ingelheim have phase 2 assets which are focusing on the neutrophil elastase inhibitor. So this is further round the vicious vortex. And so they're trying to inhibit the inflammatory responses with these neutrophil elastase inhibitor drugs. Very interestingly, from the Insmed Willow study, which is a phase 2 trial that's been completed, their drug was significantly less effective with Pseudomonas colonized patients, which were between 33 and 38% in different arms of the study. So we believe this is a really good indication that there is a real need for a specialist drug that will break the cycle earlier and stop these Pseudomonas toxins from generating the inflammatory response in the first place. So Infex, with our partner Shionogi, as Vicky mentioned, Japanese major pharmaceutical company, is recruiting now in phase 2A. With the that you see there, just a summary of what Colm and Vicky has said already. But I think what I'd also like to bring out here is that there are significant expansion options for Infex into other chronic respiratory diseases such as CF, COPD, and asthma, as well as other systemic and acute Pseudomonas infections, such as hospital-acquired or ventilator-acquired pneumonia and sepsis, for example. And Infex is already a member of the UK CF Cystic Fibrosis AMR Syndicate, working with the Cystic Fibrosis Trust to develop target product profiles for a drug such as RESP-X that might be effective in cystic fibrosis patients, as well as non-CF bronchiectasis patients. The next slide, please. We mentioned the partnership with Shionogi. We have an outstanding working relationship with Shionogi, and Vicky already displayed some data obtained by their scientists showing the the imaging of the reduction of the exotoxins in lung cells in mice. So we have an out-license agreement with Shionogi that on the completion of phase 2, they have the option to acquire an exclusive license worldwide to further develop and commercialize RESP-X. On execution of the license, we will receive from Shionogi 3 times the development costs that we've invested in the program by that date. And then when commercial sales are achieved, we will receive tiered royalties of 8 to 15% on the worldwide sales of the RESP-X drug. Next slide, please. So looking at the program overall, it's a multi-billion-dollar market, NCFB. Pseudomonas colonization at around the 25% level is a major risk factor in the progression of that disease, hospitalization, and mortality. The RESP-X is not an antibiotic, it's an antivirulence antibody that disables the key virulence mechanism of Pseudomonas that is used with these toxins to destroy white blood cells and also lung cells. And so we're aiming to prevent inflammation, disrupt the vicious vortex, and really improve the lives of people colonized with Pseudomonas in NCFB. We have very, very good preclinical data, and the imaging work brilliantly determines the mechanism of action. And we've already established safety and an excellent pharmacokinetics with a long half-life in phase 1 clinical trials. We're now recruiting in phase 2, and hopefully we'll start dosing patients in the next few weeks. And that study will hopefully be completed within the second half of 2024, so later this year. And then finally, we believe it's an exciting opportunity for investors in Infex and our current shareholders to benefit from this exciting program and the milestones that we would anticipate receiving from Shionogi, and ultimately the royalties on the commercial sales. So next slide, please. So I just wanted to finish off by telling you a little more about Infex's wider pipeline and our fundraising plans for the rest of the year. Next slide, please. So we've been talking about our lead program, RESP-X, at the top of this pipeline chart. We have 2 more drugs, MET-X, which is an AMR-resistant bypass drug, breaks a key resistance mechanism called MBL in a certain type of superbug, and allows antibiotics to continue working. And the next one after that is COVEX, which is a pan-coronavirus antiviral drug for pandemic preparedness and potential treatment for prevention of long COVID. And both of those programs we'd anticipate entering into phase 1 clinical trials during 2024. So a very exciting time for our team to bring those into clinical trials. And one latest piece of news on the COVEX program, just before Christmas, we signed a contract with the US National Institute of Allergy and Infectious Disease, NIAID, and they're providing free of charge facilities and subcontract facilities to take the COVEX asset through to be clinical ready at no cost to Infex. What I'd also say from this slide is please note that every program that we have has got significant grant funding from the ICON, the Infection Innovation Consortium, of which we're a member, Innovate UK, the Boston-based CARB-X, which is a global AMR accelerator, and also that we have an exciting pipeline of preclinical assets that will be the clinical development programs of the future. So the next slide, please. So we will be raising more money later in the year, and we think this is an excellent opportunity for new investors as well as our existing shareholders to benefit from delivery of some key milestones in the relatively short term. So on COVAX, executing further funding agreements with NIAID and other potential partners. On our MET-X asset, we anticipate generating multiple pharma licenses from different regional deals across this year and into next year. And then achieving the phase 2 milestone with Shionogi, which would trigger the license payments that we've already talked about. So we will be contacting people as we go through to the end of this quarter and into the next quarter to gauge interest in investment in Infex. But we would anticipate that a decent amount of the overall funding requirement for the next 3 years will continue to be met by additional non-dilutive grant funding and also potential partner revenues. So next slide, please. So thank you so much for your attention so far. And I would now like to bring Carl in, who's been collating any questions that have come in that you'd like to put to me or to Vicky or to Colm. Carl, over to you.
Great, thank you very much, Pete. And thanks to everyone who's submitted a question to be answered by the panel. The first question is for Vicky. Does RESP-X have any synergistic effect with traditional antibiotics?
Thanks, Carl. Yeah, it's a good question. So the way we see RESP-X being used, it will be as a maintenance therapeutic, so part of the sort of therapies that these patients receive, really to reduce exacerbations in this patient group. But we do expect that if patients experience an exacerbation, then they will, of course, be given antibiotics should they need them. So as part of the preclinical package of data, we've got infection model data where we've co-dosed the animals with both RESP-X and a variety of anti-pseudomonal antibiotics. And really what we see is that RESP-X enhances the reduction in bacterial load in the animals, and it also enhances survival of the animals as well. So it does appear that RESP-X does act somewhat synergistically with anti-pseudomonal antibiotics, at least in preclinical infection models. So we do hope that will translate through to patients as well.
Thank you, Vicky. I have another question for you. Is the activity of RESP-X affected by antimicrobial resistance?
Okay, yes. So I think I touched a little bit on this, but it's a really important question. So as we mentioned, RESP-X is not an antibiotic. So from all the in vitro testing that we've done and looking at the isolates that we've used in infection models, there is no correlation between the activity of RESP-X and the antibiotic resistant status of the organisms. So, you know, long story cut short, no, RESP-X activity is not affected by AMR. So we've profiled a lot of organisms and determined their resistance mechanisms which they carry, and there's no correlation or activity of those mechanisms on RESP-X.
Great, thank you, Vicky. The next question is, have you looked into the number of patients required in phase 2A for statistical significance and on what basis? Perhaps that'd be a good one for you, Colm.
Yeah, sure. No, another good question. Apologies if it didn't come across on the presentation. So the phase 2A, of course, is the healthy control, sorry, the patient safety and pharmacokinetic studies. So that just has 12 patients to document that the drug gets into the lung with bronchoalveolar lavage and also document the pharmacokinetics and safety. So I think the question probably refers more to the phase 2b study, which would be the randomized controlled trial, where we would be giving 4 doses of RESP-X at 3-monthly intervals with the aim to reduce the number of exacerbations. And so there's been both in-house and independent out-of-house statistics using what we know about incidence of exacerbations from previous trials, from registries, incidence of Pseudomonas colonization, etc. And we're confident that 76 patients in the phase 2b, even allowing for dropouts, that's 38 patients in each arm, 38 in placebo, 38 in the active treatment arm, is enough to show a clinically meaningful reduction in exacerbation rates. And these numbers are also consistent with the powering of recent trials in non-cystic fibrosis bronchiectasis, as well as planned trials in NCFB based on a conservative estimate of the placebo exacerbation rate. And so we're confident that, 76 patients is enough for the phase 2b randomized control trial.
Great, thank you, Colm. I've got another question that I think would be best for you as well, Colm. Is there potential to develop RESP-X in additional indications other than NCFB?
Yeah, I think again, Pete touched on this in his talk, and Vicky did as well, sort of indirectly. There's no doubt that Pseudomonas infection is very important microbe in not just non-cystic fibrosis bronchiectasis, but also cystic fibrosis. It's a really important contributor to hospital mortality in a range of situations, be that ventilator or pneumonia or hospital-acquired pneumonia, also sepsis and complicated urinary tract infection, and also severe, wound infections and burns infections. And there's a lot of interest from around the world in what RESP-X could do beyond NCFB, and various people, including the director of a Wellcome Trust-funded research network in Asia, is very keen that at some point in the future that we start looking at RESP-X with them in acute infection situations. So I think there's really quite a wide range of conditions, and I should have said also chronic obstructive pulmonary disease and asthma, also a percentage of those have issues around Pseudomonas. And so yes, lots of scope beyond non-cystic fibrosis bronchiectasis.
Great, thanks, Colm. Another question's come through for Vicky and Colm. How could RESP-X be used for acute Pseudomonas infections like sepsis that was mentioned?
Yeah, I'll pick this one up. So yeah, we did mention acute lung infection and gut-derived sepsis in particular, as we have preclinical data to support use in those indications. So we have a lot of data to support the use of RESP-X as a preventative for acute pneumonia. So Pseudomonas is particularly problematic for patients undergoing mechanical ventilation. So we have some really nice efficacy data for RESP-X showing that it's protective in this situation. And we also have data from a gut-derived sepsis model. So particularly in ICU settings, the gut really acts as a reservoir for Pseudomonas to cause systemic disease and sepsis in these patients, which can be really problematic with a high mortality rate. So similar to the chronic infection model I showed in the lung, we have some really nice data showing that RESP-X is protective from that gut-derived sepsis in mouse. So certainly plenty of preclinical supportive data to expand RESP-X into further indications.
Smashing, thank you. Just one more question that's come in, and the final one, unless anyone has any other questions to ask. Where is the phase 2 trial going to take place?
Yeah, so the phase 2b trial that we've talked about will be based in the UK. So we think based on numbers of patients and interest, at the moment we have 5 main centres. There's a 6th centre wants to come on board as well. And we know there are various satellite centres for each of those large bronchiectasis sort of programs that will be keen to take part as well. So phase 2B will be in the UK, but there is a lot of international interest both in US and Europe and Ireland and Japan for further trial options worldwide. But we're starting off in the UK.
Great, thanks, Colin. That's it for questions now, unless anyone else has any other questions. I'll hand back to Pete, and thanks again for all the questions.
Great, thank you, Carl, and thanks in particular Vicky and Colin for providing some great answers there. So just leaves me to say really thank you for taking the time out to join us today. The recording of this will also be made available by our colleagues at Walbrook and Carl at Infex. So it just leaves me finally to say thank you for Colin and Vicky for the great presentation, Carl for handling the Q&A, Louis at Walbrook for setting all this up for us, and of course you for coming along and listening to what we hope was a story about an exciting drug that we hope will do great things for patients suffering from this really terrible disease. So thanks everyone, and have a good rest of your day.